mitosis. So today we will expand that concept and look at meiosis. Now, meiosis is a process that has evolved because of sexual reproduction, and it only occurs in the reproductive cells, the male sperm and the female egg. And meiosis results in the formation of gametes. So it's just a regular extension of mitosis. The same sort of paradigm exists where you have cells divide, but in this case, you have a reduction in the number of chromosomes. So here, sort of put in sort of graphic form, a little sense of here, we have a cell which has a diploid number of 2n, the number of chromosomes are 2. So here you have here an interface here. You go through mitosis, prophase. You know here an interface, the chromosomes divide, they multiply, and they duplicate. And these are called sister chromatids. So here you have the sister chromatids, and you go through the process of metaphase, anaphase, telophase. This is simple mitosis. But in meiosis, you have a further division where the reduction division takes place, and you go into the second prophase 2, and you have these two cells here. So you have this, you have homologous chromosomes separating here, and then you go through the metaphase 2, anaphase 2, and the formation of the gametes. I did a little trick here, and when the chromosomes separate homologous, you could have at this stage here some crossing over takes place to give you a little further division of the chromosomes here, get more quality here. So here you have prophase 2, homologous chromosomes separate, and then I did a little trick here for you. In this one here, I showed some crossing over. So if you notice here, you have half of the side of one chromatid is blue and it's red, and this, so you have a little bit more variety here. This one I did it normally, so you don't have that here. And here you have the gamete formation. You have a pure N here and a pure N here. Here you have where crossing over took place. You have half of red, blue, give you a little bit more variety. And now you have this gamete formation. And this is down the divisions N. So you go from 2 to only 1 in each gamete. Chromosomes have reduced to the N number, which is called the haploid number. Remember, this 2N here was diploid, this is haploid. Now, these gametes could then form with, if it's male or female, they could now form with a female or male gamete to give you back the original 2N number. But you increase the variety and the genetic diversity in the organism. So meiosis is reduction division that increases the diversity of the genetic information that's carried in each cell. And that is the sort of advantage of meiosis or sexual, the advantage of meiosis in contributing to the genetic increase in genetic information among organisms. So sexual reproduction gives you more variety in the opposed to ordinary mitosis, which, as you know, is one could refer to that as cloning. You're getting the same number of and kind of genetic information in each generation. Here, you're increasing In some cases, even the complexity, but you have more diversity. And that is the advantage of sexual reproduction over asexual reproduction. You're getting increased diversity. And one of the tenets in biology is the more diverse you are, is the more complex you are, because then the organism is able to react to changing environmental conditions. Okay, and I think pretty much you're familiar with it. So this is the advantage of meiosis in sexual reproduction. I think I give you over. I don't know how much, but you'll be able to tell me. I don't know how much you go into this in terms of the mechanisms and the importance of meiosis in the 8th grade. But mechanically, the only difference is here, you have a second mitotic division, which contributes to the reduction division here as opposed to mitosis. So I will stop at that moment and answer any questions and see how the activity, the materials that you got were able to sort of play with it and show and see how you were able to manipulate the yarn to show some of the mechanics of this. Any questions? You're getting any questions? Could they hear me? Any questions out there? Comments? Yes, I can hear you. Thanks, Valerie. How let me ask a question. How deep, when you do this with the 8th grade students, do you go into some of the complexities? I just showed how you get some crossing over here, but that's another way to increase the variation of the genetic information in the cells. Do teachers go a lot into that and show some of that mechanism? Or you just concentrate on the... Well, that's good, yes. Well, that's good to hear, thanks. Let me just make one quick comment before I get some more comments here. I just want to clarify one. I know most of you know this, but let me just clarify here. When the gametes are formed, notice I didn't say these are cells. These diploid, these haploid gametes that are formed, they, in the true sense, they are not viable cells as you might take a look at an ordinary cell. What these have to do, these have to combine with the male or female gametes to form a viable cell so that you get back the original diploid number. So these, in fact, in female reproduction, notice we have here in this example, you have 4 haploid cells, or I say cells, but gametes that are formed. Normally what happens, at least in human sexual reproduction, 4 of these self-abort, and you only have 1 of these that then will combine with the male sperm to get back the original or the diploid number, which in the case of humans, as you guys know, is 46. So if I were doing this with humans, each one of these would have just 23, or half the number of the full complement of 46 in human populations. But these are not viable cells. These cells will self-abort if they don't get fertilized, or if they don't get fertilized with a male or female gamete. So in a true sense, although we call them cells, they're not truly cells. Okay? So keep that in mind. Okay. Pipe cleaners are good to use. Yeah.
Right now, it's kicking me out.
Okay.
So you're going to have
Okay.
to
I see here on my I do think that using yarn, etc., does help the kids understand. I do cover crossing over, but only after they understand meiosis in general. That's a good point. That's great. Pipe cleaners are good to use in place of string or yarn. Yes, I am a firm believer in anything. Oh, you unmute me?
Go ahead.
Okay, could you guys hear me now? Yeah, as I was saying, I think that the activity is a great way. If you have your kids do this as a project, or they come together in a sort of community to show some aspects of this mechanical process, I think it's a good way that as you listen to them go through and talk through the activity, you could discover a lot of misconceptions that some of them may have after your presentation. It's a good way to sort of clarify that using the yarn, or as someone says, cleaners and so on that you can use. So anything that will help you get a sense of how the kids are processing this information is a good way to discover and clear up any type of ideas that they may have. Hello, any other questions or comments? Okay. Well, if there are no questions, no comments, I thank you very much. I hope this has given you a good sense of what you can do in your class, whether you use yarn or string or any other type of manipulative to get across the idea of meiosis. Usually, and I know according to the standards, the kids were coming to you knowing mitosis, and as they get a chance to review that, is a good way to introduce meiosis to them and make sure that they see the difference between meiosis and mitosis. And keep in mind that this only takes place in the reproductive cells as opposed to the other cells of the body, the somatic cells where you have ordinary self-division. So mitosis is essentially for growth and repair of these somatic cells in our bodies, or in most living, in sexual reproducing organs. But meiosis is clearly limited to the reproductive cells, the male sperm and the female egg. Thank you very much. Have a great summer. Stay safe.